Hello everyone, welcome to my first biochemistry live lecture. For the topic today, we will talk about proteins. Let's start with the objective of medical biochemistry. This year, you will enter the world of biochemistry, the science that reveals not only how the human body functions at the molecular level, but also how, through understanding biochemical processes, you will be able to explain the foundations of nutrition, health, and longevity, key topics of modern preventive medicine. We are together with students from Medical University of Plovdiv. The topics for today will be peptides, polypeptides, and proteins. We'll talk about amino acids as structural units of proteins and levels of organization of protein molecules, but only the first three levels will be for the first lecture. Let's start with a definition before about the proteins. What are these molecules? Actually, the proteins are macromolecules that consist of amino acids which are linked together by peptide covalent bonds. They can be arranged in one or more polypeptide chains. And one more about proteins. The proteins molecule are the most abundant macromolecules in our body, unlike all the rest, carbohydrates and lipids. And I very like to say that they are the most privileged molecules because they are produced by one pathway from DNA, one information pathway from DNA, mRNA, and proteins. Actually, DNA is responsible for the production only of proteins, but not carbohydrates and lipids. That's why if we need to find somewhere some defect in proteins, we need to find it in DNA as DNA mutation or something else. They have a variety of functions, and as you see, the last function is energy production. The last, because the first function of energy production is about the carbohydrates and lipids, but not proteins. Let's start from the beginning to talk about functions of proteins. The first function is structural function. You need to know the corresponding examples. Structural function refers to the main tissue with such a dysfunction, which is connective tissue, of course. And examples are collagen, elastin, keratin, but not only from the connective tissue. Actually, there are many other proteins that have such a structural function, like in ribosomes. Because ribosomes consist of not only RNA molecules, rRNA, but also plus proteins. Also chromatin. Chromatin is a structure with DNA plus proteins too. Number two, catalytic function. Catalytic function, of course, all enzymes, with one exception, but because here the question is, are all enzymes proteins or not? Actually, not all enzymes are proteins. We have some examples of enzymes that are not proteins, and these are called ribozymes with RNA in the structure. But when we're talking about enzymes in your education, we'll talk about protein enzymes, not ribozymes. Transport function. Transport, students, why we need to know all the time about the transport? We will ask yourself all the time, is this molecule possible to be transported alone or not? It depends on if the solution is water soluble or not. Actually, in our body, we have two places with water soluble solution, and these are blood and cytosol. And one important place with lipid soluble environment, and these are all cell membranes, not only cell, all membranes. That's why if some water-soluble compounds need to be transported in the blood or cytosol, no problem. They can be transported alone without any transporters. But if they need to be transported through the membranes, they cannot be transported through the membranes. They need to be transported thanks to specific transport proteins. Remember, no any other transport compounds except proteins. They are the only one that can be transported all lipid-soluble compounds. No carbohydrate transporters, no lipid transporters. They are only protein transporters. They can be located in the membranes like different carrier proteins, or like these examples in the blood, like hemoglobin. Hemoglobin is a transport protein in the blood which transports oxygen from the lungs to the tissues and carbon dioxide from tissues to the lungs. One more example, transferrin, which transports iron 3+ in the blood. And one more, it's albumin, which is absolutely classical example of transport protein, which is non-specific and can transport many compounds like fatty acids, hormones, calcium, bilirubin, and etc. One more. If we're talking about the blood, you understood about the specific transporters in the blood. If we're talking about the membrane, again we need specific transporters which are located in the membrane. Which compounds in general are water soluble? Actually, proteins and carbohydrates. Lipids are not. And all derivatives, of course. A regulatory function, it's about hormones, like, but because today we're talking about only proteins, these are protein hormones. Do you know some examples of protein hormones? Yes?
Endorphin.
Okay, it's a peptide, insulin, glucagon, vasopressin, oxytocin. Yes, oxytocin. Okay. A receptor and signaling. Don't forget growth factors. Growth factors are some polypeptides that have regulatory function too. A receptor and signaling. Here you need to remember that all, absolutely all, receptors are proteins, no any lipids or carbohydrate receptors. They are proteins, which means the same. If there is some defect in any of receptors, we need to find it in DNA, again in DNA, because from DNA we can take the information for the synthesis of any of proteins. Defense function, it's about antibodies. The other name immunoglobulins. They are proteins too. Clotting factors are used in the blood coagulation cascade. This is the defense from bleeding. Motor or contractile function, it's about muscle tissue with actin, myosin, troponin, and tropomyosin. Storage function, it's about one protein. It's a very good example. It's a ferritin, which is used for the storage of oxygen in the muscles, and myoglobin for the storage of iron, about ferritin, and storage of oxygen, about myoglobin. And the last function is energy production. When we rely on proteins for energy production, only in long-term starvation, students. In the beginning, we need for energy carbohydrates. We prefer carbohydrate or lipids. But the proteins, you can see how many functions they have. That's why this will be the last function. molecular variants of proteins. Actually, there are different types of proteins according to their location. They can be isoproteins first. Isoproteins means the same protein with the same function, but with different localization in the same individual. Particularly if we're talking about enzymes, they are called isoenzymes. For example, muscle or heart creatine kinase means that if we test high level of heart creatine kinase in the blood, this means maybe disorder in the heart, which can be myocardial infarction. Or if we test high level of muscle creatine kinase in the blood, this maybe means some disorder in the muscles. That's why the isoenzymes are very important for clinical diagnostics. Other examples are about the organelle localization in the same cell, like mitochondria and cytosolic alanine aminotransferase. The same cell, the same enzyme, but in different localization in different organelles. The alloproteins are located in different individuals of the same species, and they refer to existing of gene polymorphism. The example is about blood group antigens, Rh factor, and etc. Heteroproteins. These protein students are located in different species, which means, example, animal albumin, human albumin. You understood that the function of albumin is transport in the blood. Absolutely the same function there is in the animal and in the humans. But we cannot take animal albumin in us, in our blood. Why not? Because the foreign proteins have foreign antigen determinants, which means our immune system will destroy it. That's why we cannot accept animal albumin in our blood directly. The size. According to the size, we have three different types. The first ones are called oligopeptides. Oligopeptide students consist of 2 to 10, 20 amino acids according to different authors, and they can be 2 amino acids, which means they will be called dipeptides, 3 amino acids, tripeptides, and etc. From 20 to 100 amino acids, they are called polypeptides, and over 100 amino acids, they will be proteins. Let's discuss about examples. First, glutathione. What is this, a glutathione? You can see that glutathione exists in two forms. The first form of glutathione is called reduced glutathione. The abbreviation, remember the abbreviation, is GSH. It's a tripeptide because consists of 3 amino acids. Do you know which is the pink one? It's a glutamate. The blue one, cysteine, and the red one is glycine. The most important is this sulfhydryl group of cysteine, which that's why the glutathione is called reduced glutathione. After the combination of 2 molecules of glutathione is formed oxidized form of glutathione. Students, actually here is written, this is the master intracellular antioxidant. What is the role of this compound? The role is that glutathione, this one, reduced form, is used as a cofactor of the enzyme glutathione peroxidase. This enzyme is used for the detoxification of reactive oxygen species, and especially in this situation with the enzyme, I'm talking about hydrogen peroxide. This reactive oxygen radical is not radical, is that's why the total name is species, need to be detoxified by this enzyme glutathione peroxidase. The cofactor is glutathione. Actually, in our organism are produced all the time reactive oxygen species and some other reactive species that need to be detoxified thanks to antioxidant enzymes. One of them is this one that I said. Another examples of oligopeptides, antidiuretic hormone or vasopressin, and consists of 9 amino acids, and oxytocin 9 amino acids too. Do you know where is the place of synthesis? Where are synthesized? In the okay, but exactly where? In the hypothalamus. They are produced in the hypothalamus they are saved in the pituitary gland, in the posterior part of the pituitary gland. Other example are gastrin, cholecystokinin, there are different hormones. Polypeptide. The first example is insulin. 51 amino acids. Let's see the figure. You can see, students, 2 polypeptide chains. A chain, 21 amino acids, and B chain, 30 amino acids. What are these? This one, and this one? Yes? Bonds. What kind of bonds? Disulfide bonds or disulfide bridges. Actually, you can see that there are 2 types of disulfide bonds. This one and this one are called interchain. This one is called intra-chain disulfide bond. But I would like to explain something else about this molecule. Most often in our body, some of the proteins are produced not directly in the active form. They are produced in the prof form in the beginning. It's the chain A. This is the chain B of insulin. And these are both of the disulfide bridges between A and B, but this one is called C chain. In the process of synthesis of insulin, by the way, where is the synthesis of insulin? In which cells? Yes?
In the pancreatic
in pancreas, but it's an anatomy answer. Tell me the biochemistry answer, which means cells.
B cells.
Yes, beta cells. Yes. In the beta cells of pancreas is produced insulin. This is the proinsulin. And after the maturation and removing of this one, C-peptide is removed. C-peptide, which can be tested in the clinical laboratory, by the way. At the same time is produced A plus B, which is insulin. This one is insulin. Insulin. Which means, if you know how many is C-peptide, you will understand how many is insulin. It's very important test in the clinical laboratory to know how many is C-peptide. Do you know what is the main function of insulin? It is used for Yes?
To decrease sugar levels in the blood.
Yes. Yes, leads to decrease of glucose in the blood, because sugar, yes, in English is used the term sugar, but sugar actually means sucrose, which means glucose plus fructose. That's why to be punctual, use glucose level. This is the only hormone that decreases the glucose in the blood, the only one. All the rest are contrainsular hormones, which means they increase the glucose in the blood. One more example of synthesis of proteins as proforms. This was one of the examples about insulin, but there are many other examples about the digestive enzymes, students. Digestive enzymes are produced as proforms too. Otherwise, they can be digest the place, the organ of synthesis. Tell me, do you know where is the place of synthesis of most of the digestive enzymes? In which organ? Yes?
Duodenum.
The duodenum, the small intestines, here is the places of action, but not the places of synthesis. Where is the place of synthesis, yes?
I think stomach.
Stomach is the first one, which is called pepsin. Pepsin, which is produced from pepsinogen. Pepsinogen is the precursor. Pepsin active. But it's only one in the stomach. All the rest are in small intestines, but are produced from pancreas. Pancreas. In the pancreas, there are two parts. Endocrine part with beta cells, with alpha cells, delta cells, and endocrine part that produces the digestive enzymes. If there is self-activation of the digestive enzymes in the pancreas, there will be digest the pancreas. It is one of the very, very big pain in medicine, which is called acute pancreatitis. Okay, trypsinogen, trypsin is an example that is produced in the pancreas, but goes to the small intestines, where is the place of action. What is produced by alpha cells, do you know? They produce glucagon. What is the action? As I said before, opposite of insulin, which means they increase, the glucagon increases the glucose in the blood. And this was the next example of polypeptides. It consists of 29 amino acids. And over 100 are proteins. In general, proteins are water soluble, but with some details about the amino acids. Amino acids, the monomers of proteins. Protein student, actually amino acids are, some authors say that there are more than 300, but only 20 of all these amino acids are included in our proteins in the human body. 20 amino acids specified by genetic code that are encoded in DNA. According to if they are included in proteins or not, they are called proteinogenic. All these 20 are proteinogenic. Except glycine, all the rest are asymmetric and exist as L enantiomers or L forms, which means the amino group is located on the left of C alpha carbon. If the amino group is located on the right, it will be the amino acid. The question is, do you know, are there the amino acids in our organism? Actually, in the proteins, no, but in the body, yes. Some of them, for example, D-aspartate, D-serine, these are neurotransmitters. They exist alone, not in the proteins, only alone. We have enzyme that can switch these forms of amino acids from L to D. They are called racemases, these enzymes. At physiological pH, approximately 7.4, amino acid exists predominantly with sweeter ion. The amino group is protonated, is NH3+, and the carboxyl group is deprotonated and is COO-. Except that we have one hydrogen atom and a radical. The first classification of amino acids is according to the nutritional requirements. You need to know by heart. I won't recommend you to learn anything by heart, because in biochemistry there is absolutely logic everywhere, but you need to know by heart all which are essential amino acids. What is this essential? Do you know? What does it mean, essential? Important, but something else there is here. They cannot be synthesized in the body. Cannot. They can be taken only by diet. Yes, only by the diet, which means all the time we need to think about if my food contains essential amino acids or no. You need to plan your food to contain essential amino acids. Which are they? They are valine, leucine, isoleucine, lysine, methionine, phenylalanine, threonine, and tryptophan, 8, plus 2 more, arginine and histidine, which are semi-essential, but according to many authors, they are absolutely essential for many people, not only children. And only the half can be synthesized in our organism. They are called non-essential. Which kind of food contains amino acids? Yes?
Meat, eggs.
Yes, meat, eggs, fish.
Fish. Some
Nuts.
nuts.
Beans. Yes, from plants. Yes, all this food contain amino acids. What does it mean, high quality protein food? This means that contains all essential amino acids. This means high quality. It's not enough to have a label on the food. High protein bar, or high protein skyr, or high protein something else. You don't need only high protein, you need enough protein food, not high. You need to have all essential amino acids, but not to be too much that your recommended dose. The next classification is according to the radical. And the amino acids are differ. The first types are called aliphatic amino acids. These are glycine, alanine, valine, leucine, and isoleucine. Actually, students, valine, leucine, and isoleucine are called also branched chain amino acids that are used in the muscles. Hydroxy-containing amino acids are serine and threonine with hydroxyl group that is very important in the processes of post-translational modification of proteins. What does it mean, post-translational? Means that this group can be modified with adding of one phosphate atom and removing of one phosphate atom, which is called phosphorylation, dephosphorylation, for this process, we need these amino acids. Sulfur-containing amino acids, as cysteine that we said about disulfide bridges. Methionine is the second one with sulfur. Proline is the only one which is cyclic amino acid with secondary amino group, this one. And aromatic amino acids, which are phenylalanine, tyrosine, and tryptophan. Amino acid containing conditional basic group, which means additional amino group. And these lysine, arginine, and histidine actually are they have positive charge. They are charged amino acid because of the more positive charges thanks to the amino group. These two amino acid aspartate and glutamate, they contain additional carboxyl group. That's why they have negative charge. And this one, asparagine and glutamine, are their amides. They have additional amino group but not charged. Just one more nitrogen atom there is here. This is another classification that differ amino acids according to their charge. Actually, students, approximately half of the amino acids are polar, which means more water soluble, and non-polar, which means less water soluble. These amino acids that are non-polar are located mainly in the inside, in the internal part of the proteins. Polar mainly on the surface of the proteins. The non-polar are all aliphatic, glycine, alanine, valine, leucine, and isoleucine, plus methionine and proline, plus this aromatic. Actually, tyrosine is, some authors say that this is polar, some it's not polar. Actually, more of them think about that it is polar amino acid because of the hydroxyl group of the tyrosine. And these polar are two types, uncharged and charged, positively and negatively, that I said before in the previous slide. What kind of compounds are these ones? According to their chemical structure, can you understand? Can you see? It is a carboxyl group, amino group, carboxyl amino group. They are called amino acids. They are amino acids, but are they encoded in DNA? Do you know these amino acids? No. They are not encoded in DNA. They are produced by two ways. First, by post-translational modification. Example, from proline, hydroxyproline. From lysine, hydroxylysine. From glutamate, gamma-carboxyglutamate. And during metabolic processes like ornithine and homocysteine. Remember the first two that are included in collagen, students, because they are very important in the structure of collagen. Let's start talking about the levels of organization of proteins. For this first lecture, we will talk about only first three levels of organization: primary structure, secondary, and tertiary. First, primary structure. What is this primary structure? It refers to number, type, and sequence of amino acids that are linking together by peptide covalent bonds. Peptide covalent bonds are formed between two neighboring amino acids, the carboxyl group of the first one and the amino group of the second one, after the process of removing of one molecule of water. And you can see here the primary structure. This is the process of condensation between these two groups and formation of one new bond called peptide bond. These atoms, carbon, oxygen, nitrogen, and hydrogen atoms, CO and NH, are formed together so-called peptide group. Actually, this is the example of one dipeptide, because it consists of only 2 amino acids. And in all peptides, never mind that is dipeptide, tripeptide, in non-polypeptide chains, we have 2 terminals, N-terminal or nitrogenous end, and C-terminal or carboxyl end, or carboxyl terminal. The key characteristics of peptide bonds are that this bond is strong covalent bond with resonance. That's why can be also named like partial double bond character. Actually, this bond is shorter than the single one. You can see this here. This is the peptide bond between carbon atom and nitrogen atom. It is shorter. And one more, all these 6 atoms, CO, NH, and these 2 C alpha atoms from the adjacent amino acids, they are located in one plane. All of them are in one plane. That's why we can say that this is the planar peptide group. And that's why, because of the rigidity, because of the planar structure, no possibility of rotation between C and N atom. Possible rotation can be only in this angle between C and C alpha, which is called here, you can see phi angle, psi angle, this one psi angle, and this one between C alpha and N, which is called phi angle. Only here there is free rotation. About the configuration of radicals of polypeptide chain, they are located in transposition, which means the first radical is here, the second here, here, here. This is the to prevent interfering between radicals. In physiologic pH, the peptide bond is uncharged. It's not ionic, it's a polar, because the nitrogen atoms, these nitrogen atoms has partial positive and the oxygen atom negative charge. One more important about peptide bonds. Peptide bond is used for the formation of primary structure. This is the main bond. The basic bond. But thanks to this bond is formed the secondary structure, because this peptide bond is used for the formation of another bonds called hydrogen bonds. Hydrogen bonds, which are critical for the formation of secondary structure. What is this secondary structure? Secondary structure, students, means 2 main types, thanks to hydrogen bonds, as I said, and formation of number 1 are alpha helix, which means folding, some folding, but planar folding of polypeptide chains, thanks to the formation of hydrogen bonds, which are located according to the axis located hydrogen bonds. This is the first type. Second type are called beta sheets. The beta sheet can be 2 types. Here you can see the alpha helix. The radicals of the polypeptide chain are located outside the chain, outside the axis. But beta sheets, beta pleated sheets, can be 2 types. They can be with parallel polypeptide chains, which means more than one polypeptide chain, and hydrogen bonds are located perpendicular to the axis, and second types of beta sheet, when we have anti-parallel formation of beta sheet, and again the hydrogen bonds are located perpendicular to the axis, the same. And it's a planar structure, no 3-dimensional structure. there are, except alpha and beta helices, there are some ordered regions which are called loops. And maybe 50% of the regions are ordered. Loops are disordered segments, that connect the alpha and beta helices, but they are very important for the signal transduction, actually. This is the process of the transmission of information of hormones from outside the cell to inside the cell. And after that, there is formation of supersecondary structure, which means formation of motifs. Motifs means combination of helix turn helix, helix loop helix, or beta alpha beta motif. For example, alpha loop alpha loop, beta beta loop beta beta loop alpha. It's beta beta alpha alpha. It's a complex motif, or simple motif, alpha alpha beta beta. The motifs are used, this binding of thanks to the loops is used mainly in protein-protein interactions when we're talking about the transcriptional factors. One more, remember that motifs are structures in the secondary structure. They are not tertiary structure. Tertiary structure. For the first time, we're talking about three-dimensional structure, which means that there is additional folding of protein into space and formation of something new, which is shape. For the first time, we have shape, which are two types of shape, globular and fibrous proteins. And students, is it obligatory all proteins to have quaternary structure? No, of course not. Tertiary structure can be the last. Except shape, they have specific function. And during the formation of this three-dimensional structure are formed specific other structures that are called domains. Domain means, for example, this one. Sometimes it's not obligatory to have domains in all proteins with tertiary structure, but sometimes they have domains. For example, this domain is responsible for the catalytic function. This domain is responsible for the activation with a regulatory function, or etc. And remember, students, this contact between domains is covalent. It's covalent, obligatory, covalent. Which are the interactions of tertiary structure? Here, the first important bond which stabilizes the tertiary structure is disulfide bond. You know about disulfide bond. It's a combination between two cysteine residues to form one new compound, which is called cysteine, no cysteine, cysteine, by dehydrogenation. The second one are hydrophobic interactions between hydrophobic amino acid residues, polar interaction between polar amino acid residues, which can be hydrogen bonds, but not hydrogen bonds that are formed between peptide bonds that are used in the secondary structure. These bonds are formed between radicals of amino acids. This is the difference. Number two, ionic interactions. For example, between carboxyl group of from some amino acids like aspartate, glutamate, which have additional carboxyl group, and the amino group of lysine, plus, minus, and ion dipole interactions. This table shows the different shapes of proteins according to their tertiary structure, and the most important from here is to understand that fibrous proteins are insoluble or less insoluble in water, and the function is in the structure of the body. And examples are keratin, collagen, elastin. According to the globular proteins, the most important is to understand that they are polar, water-soluble compounds, and they have many functions. Enzymes, obligatory, are globular. Hormones are globular proteins. Transport proteins are globular, and etc. Two specific examples of one fibrous and one globular proteins. Remember, students, that the collagen amino acid sequence is glycine XY, glycine XY, N, glycine XY. This is repeated many times to form, not here, approximately 1,000 amino acids. Actually, the form of collagen is 3 alpha helices, 3 polypeptide chains that form that are alpha helices, and they contain this sequence. Every third amino acid is a glycine, which are X and Y. Do you remember I said today which can be X and Y? They can be proline and hydroxyproline. Do you remember hydroxyproline, which was non-proteinogenic amino acid? Yes, and less frequently lysine and hydroxylysine. That's why the content of glycine and proline is very high. Very high. Another example is myoglobin, which is a globular protein. Do you know what is this, students? This one in red color, inside the myoglobin, what is this? Actually, this is again alpha helix, but in the shape of globular. Do you know about one molecule called heme? It's a heme. In the center of myoglobin, inside, it is called heme. Inside the heme, there is one molecule of iron 2+. Thanks to the iron, there is this possibility of myoglobin to use for the saving of oxygen and carry oxygen in the muscle tissue. Again, the myoglobin formation. And actually, students, this was the last slide for today. Thank you for attention today.